Hello and welcome, dear students. Today we are going to discuss about the topic Introduction to Ecological Genetics and Polymorphism. The main objectives of today's lecture are to know the basics of ecological genetics, to understand the phenomena of genetic polymorphism, to understand the genetic polymorphism and its relevance to evolutionary theory, to know about the genetic polymorphism and selection, to understand the importance of polymorphism as well. Dear students, let's first start with Introduction to Ecological Genetics and Polymorphism. Ecology is the study of relationships between organisms and their environments, whereas ecological genetics focuses more specially on the genetics of ecologically important traits, that is, the traits that influence ecological relationships. Therefore, ecological genetics is the branch of science that investigates the relationship between genetic change and features of the biotic, that is the living, and abiotic, that is non-living environment. Ecological genetics is the study of genetics in natural populations. Traits in a population can be observed and quantified to represent a species adapting to a changing environment. This contrasts with classical genetics, which works mostly on crosses between laboratory strains and DNA sequence analysis, which studies genes at the molecular level. Ecological genetics is an especially useful tool when studying endangered species. Metabarcoding and eDNA are used to examine the biodiversity of species in an ecosystem. For ecological genetics, in contrast, the central focus is the causal relationship between genetic variation and environmental variation, where obvious, that is especially discrete traits, phenotypic variation is assumed to have an underlying genetic component. The advent of molecular tools and the development of molecular ecology within ecological genetics has greatly broadened the conceptual scope of the field since last decade. At its inception, ecological genetics focused particularly on traits that influence fitness, such as those that affect survival and reproduction. This focus has been maintained, although ecological genetics now also investigates the ecological and evolutionary processes that influence patterns of genetic variation in natural populations. Therefore, it can also be considered a study of genetic processes associated with microevolutionary change. Although both Charles Darwin and Alfred Russel Wallace brought together ecological and genetic concepts in the 19th century, the term ecological genetics was first used by E.B. Ford in his groundbreaking book Ecological Genetics. The first geneticist to begin work in the field of ecological genetics was indeed E.B. Ford, who mostly worked in 1950s and published a book entitled Ecological Genetics in 1975 after working with Manuela Giortina. This type of evolutionary biology study was only possible after gel electrophoresis had been designed in 1937, as prior to this a high throughput method for DNA analysis did not exist. With the development of polymerase chain reaction or PCR and polyacrylamide gel or PAGE electrophoresis, this field of study began to become more popular. Genetic ecology, on the other hand, is the study of the stability and expression of varying genetic material within abiotic mediums. Genetic material has the ability to be taken up by various organisms that exist within an abiotic medium through natural transformations that may occur. Thus, the field of study focuses on interaction, exchange, and expression of genetic material that may not be shared by species had they not been in the same environment. Dear students, let's now discuss the gene transfer between organisms in an environment. Genetic information may transfer throughout an ecosystem in multiple ways. The first, which on the smallest scale being bacterial gene transfer. Bacteria have the ability to exchange DNA, and this DNA exchange or horizontal gene transfer may provide various species of bacteria with the genetic information that need to survive in an environment. This can help many bacterial species survive within an environment. A similar event has the ability to happen between plants and bacteria. For example, Agrobacterium tumefaciens has the ability to introduce genes into plants to cause the development of gall disease. This occurs throughout genetic transfer between the Agrobacterium tumefaciens and between the plant in question. In fact, a similar event occurs each time viral infections occur within living organisms. The virus, whether positive or negative sense virus, require a living organism to replicate their genes and produce more viruses. Once a virus is inside a living organism, it utilizes polymerases, ribosomes, and other biomolecules to replicate its own genetic material and to produce more viruses. Genetic material similar to the original virus. That means it makes use of the living system to reproduce itself. Thus, gene transfer may occur through many varying means. Thus, throughout each ecosystem, the study of this gene transfer, whether it be through a bacterial ecosystem or through the ecosystem of an organism, genetic ecology is the study of this gene transfer and its causes. The origins of ecological genetics are founded in the modern synthesis, where the integration of genetics and Darwinian evolution was achieved. Evolutionary genetics tend towards a branch of applied mathematics founded on Mendelian sayings. Ecological genetics is grounded in the reciprocal interaction between theory and empirical observations from field and laboratory. Most studies in ecological genetics begin with one or the other of the two most prominent patterns of nature adaptation, the fit between an organism and its environment. The maintenance of two or more phenotypic or genetic forms within a single population by natural selection. With the development of molecular ecology, the latter can be significantly extended to explore phenotypic differences between species in a phylogenetic context. This permits a more incisive investigation of the environments that favor adaptive divergence of phenotypes between species. Moreover, the same molecular methods useful in phylogenetics can often be modified to identify the genes of chromosomal regions responsible for those interspecific phenotypes. Although natural selection is the only evolutionary force that can account for adaptation, several evolutionary forces acting alone or in combination can sustain a genetic polymorphism, at least transiently. Dear students, let's now discuss the ecological genetics and genetic polymorphisms. Since all polymorphisms have a genetic basis, genetic polymorphism has a particular meaning. Genetic polymorphism is the simultaneous occurrence in the same locality of two or more discontinuous forms in such proportion that the rarest of them cannot be maintained just by recurrent mutations or immigration originally defined by Ford. The later definition by Cavell, Stronza, and Bodner is currently used. So genetic polymorphism is the occurrence in the same population of two or more alleles at one locus, each with appreciable frequency, whereas the minimum frequency is typically taken as 1%. The definition has three parts. One, sympatry, that is one interbreeding population. Two, discrete forms. And three, those that are not maintained just by mutation. The term polymorphism was originally used to describe variations in shape and form that distinguish normal individuals within a species from each other. Presently, geneticists use the term genetic polymorphism to describe the inter-individual functionality, silent differences in DNA sequences that make each human genome unique. Genetic polymorphism is actively and steadily maintained in populations by natural selection, in contrast to transient polymorphism, where a form is progressively replaced by another. Genetic polymorphism relates to a balance or equilibrium between morphs or forms. The mechanisms that conserve it are types of balancing selection. Dear students, let's now discuss the mechanism of balancing selection. Heterosis or heterozygote advantage. Heterosis, the heterozygote at a locus is fitter than either homozygote. Frequency dependent selection. The fitness of a particular phenotype is dependent on its frequency relative to other phenotypes in a given population. For example, prey switching, where rear morphs of prey are actually fitter due to predators concentrating on the more frequent morphs. Fitness varies in time and space. Fitness of a genotype may vary greatly between larval and adult stages or between parts of habitat range. Selection acts differently at different levels. The fitness of a genotype may depend on the fitness of other genotypes in the population. This covers many natural situations where the best thing to do, that is from the point of view of survival and reproduction, depends on what other members of the population are doing at the time. Now, the pleiotropism concept. Most genes have more than one effect of the phenotype of an organism, that is pleiotropism. Some of these effects may be visible and others cryptic, so it is often important to look beyond the most obvious effects of a gene to identify other effects. Cases occur where a gene affects an unimportant visible character, yet a change if fitness is recorded. In such cases, the gene's other, that is cryptic or physiological effects, may be responsible for the change in fitness. Pleiotropism is posing continual challenge for many clinical dysmorphologists in their attempt to explain birth defects which affect one or more organ systems with only a single underlying causative agent. For many pleiotropic disorders, the connection between the gene defect and the various manifestations is neither obvious nor well understood. If a neutral trait is pleiotropically linked to an advantageous one, it may emerge because of a process of natural selection. It was selected, but this doesn't mean it is an adaptation. The reason is that although it was selected, there was no selection for that particular trait. Dear students, let's now discuss epistasis. Epistasis occurs when the expression of one gene is modified by another gene. For example, gene A only shows its effect when allele B1 at another locus is present, but not if it is absent. This is one of the ways in which two or more genes may contribute to produce a coordinated change in more than one characteristic, for instance, in mimicry. Unlike the supergene, epistatic genes do not need to be closely linked or even on the same chromosome. Both pleiotropism and epistasis shows that a gene need not relate to a character in the simple manner that was once supposed. Dear students, let's now discuss the origin of supergene. Although a polymorphism can be controlled by alleles at a single locus, for example, human ABO blood groups, the more complex forms are controlled by supergenes consisting of several tightly linked genes on a single chromosome. Batesian mimicry in butterflies and heterostely in angiosperms are good examples. There is a long-standing debate as to how this situation could have arisen, and the question is not yet resolved. Whereas a gene family that is several tightly linked genes performing similar or identical functions arise by duplication of a single gene, original gene, this is usually not the case with supergenes. In a supergene, some of the constituent genes have quite distinct functions, so they must have come together under selection. This process might involve suppression of crossing over, translocation of chromosome fragments, and possibly occasional cistron duplications. That crossing over can be suppressed by selection has been known for many years now. Debate has centered around the question of whether the component gene in a supergene could have started off or separate chromosomes with subsequent reorganization, or if it is necessary for them to start on the same chromosome. Originally, it was held that chromosome rearrangement would play an important role. This explanation was accepted by E.B. Ford and incorporated into his accounts of ecological genetics. However, many believe it more likely that the gene start on the same chromosome. They argue that supergenes arose in situ. This is known as Turner's sieve hypothesis. John Maynard Smith agreed with this view in his authoritative textbook, but the question is still not definitely settled as of date. Dear students, let's now discuss the genetic polymorphism and relevance to evolutionary theory. Genetic polymorphism is defined as variations among chromosomes, alleles, or other DNA sequences. Genetic polymorphisms, whether studied in the form of allozymes, RFLP, mini and microsatellite variation, or DNA sequences, have become useful tools in a variety of research fields, such as population genetics, evolutionary genetics, systematics and molecular phylogeny, human genetics, agricultural genetics, and forensics. The first genetist to begin work in the field of ecological genetics was E.B. Ford. The significance of the work on ecological genetics is that it has shown how important selection is in the evolution of natural populations, and that selection is a much stronger force than was envisaged even by those population geneticists who believed in its importance, such as Halden and Fisher. Polymorphism was crucial to research in ecological genetics for E.B. Ford, and his co-workers who studied ecological genetics from the mid-1920s to the 1970s, and the similar type of work continues even today, especially on mimicry front. The result had a considerable effect on the mid-century evolutionary synthesis and on present evolutionary theory. The work started at a time when natural selection was largely discounted as the leading mechanism for evolution, continued throughout the middle period when civil rights ideas on drift were prominent, to the last quarter of the 20th century when ideas such as Kimura's neutral theory of molecular evolution was given much attention. In just a couple of decades, the work of Fisher, Ford, Arthur, Kane, Philip Shepherd, and Cyril Clarke promoted natural selection as the primary explanation of variation in natural populations instead of genetic drift. So evidence can be seen in Mary's famous book Animal Species and Evolution and Ford's Ecological Genetics. Kimura drew a distinction between molecular evolution, which he saw as dominated by selectively neutral mutations, and phenotypic characters, probably dominated by natural selection rather than drift. Genetic polymorphism, as defined by Ford, is the occurrence together in the same locality of two or more than two discontinuous forms of species in such populations that the rarest of them cannot be maintained merely by recurrent mutations. Ford further distinguished two types of selective polymorphisms, that is, transient polymorphism and balanced polymorphism. Transient polymorphism caused by a new favorable mutation in the process of displacing its ancestral allele by natural selection was considered unlikely because advantageous genes will usually have been already incorporated into the genetic constitution of the species. On the other hand, balanced polymorphism is a situation in which two different versions of a gene are maintained in a population of organisms because individuals carrying both these variations are better able to survive than those who have two copies of either version alone. Thus, the primary goal of the ecological genetists is to differentiate exactly how natural selection is acting to maintain a balanced polymorphism by the relative strength of opposing fitness effects acting on the different sexes at different stages in the life history, at different localities, or at different times in the lifetime of an organism. Dear students, genetic polymorphism through multiple alleles at individual loci provide a mechanism to tag a gene or a piece of DNA, providing a powerful tool for a variety of investigators. These investigations cover a wide berth of objectives, including identification of genotypes in paternity and forensic studies, tracking the movement of individuals in field studies following the progression of selection experiments in laboratory populations, mapping of quantitative loci affecting economical traits in plants and animals, mapping of disease genes in humans, and evolutionary comparisons of DNA sequences and chromosome organization between related species. The use of genetic polymorphisms are almost endless. Within a mere 50-year period, our picture of genetic variation in natural populations has moved from near monomorphism to ubiquitous poriformism in all organisms whose populations have not gone through severe bottlenecks in their recent evolutionary history. Dear students, let's now discuss the genetic polymorphism and selection. Polymorphism consists of mutations that escape DNA repair system or cell divisions. Their rate of appearance is therefore a biological variable. The considerable amount of sperm produced by male mammals means that there is much more cell division in male germ cell line than in female germ cell line. This means that in humans, the mutations are mainly produced in male lines and depend on the age of the father. Each birth produces about 100 new mutations per genome, but because only a small part of the genome is coding, 99% of them have no effect on survival or fertility because of non-coding. They are called neutral. A new allele can be neutral, harmful, or advantageous. We could think that in a genome compromising only neutral alleles, the drift of allelic frequencies would compensate for one fluctuation on the other, and that the allele diversity could remain stable in the long term. But this impression is false. Gradually, diversity is eroding. This phenomena is very similar to the loss of diversity of family names, a slow but significant phenomena in human isolates such as remote villages. When a family does not have a boy, it does not pass on its surname. The same surname can be transmitted by related families, but the smaller the population, the greater the probability of names being lost. This is obviously not due to any biological property of the Y chromosome which accompanies male births. Chance is enough to explain it. This property reflects the fact that the constitution of a daughter generation from a parental population follows the principle of a draw with replacement. Like the Y chromosome, some genes of the parental generation are not derived and are not found in a daughter population. If the genes of the progeny are randomly drawn in a population of constant size, the probability that genes are not drawn is given by a Poisson's law of parameter. These undrawn genes, that is more than a third, disappear without offspring. Their absence is compensated by parental genes which by chance have left more descendants. Now, importance of polymorphism. Natural population genetics in 1930s and 1960s wanted to assess the extent of polymorphism and discover its potential utility in terms of evolution, and they discovered an increasing number of polymorphisms in nature. Debates opposed researchers who considered that genetic diversity conferred an advantage in itself and that selection maintained it at high levels to researchers who considered that selection led to a phenotype in a finite size population. It was finally the discovery in 1966 of extremely high levels of molecular polymorphism which could not be explained by natural selection alone. That allowed the Japanese Kimura and Ota to put forward their theory. It was realized that the alternative to Darwin's theory of natural selection was not the fixity of species as thought by Darwin's opponents, but a continuous genetic change predicted by the neutral model, similar to the random walk of a diffusion phenomena in physics. This vision was definitely accepted in the 1980s. However, the very low value of the effective population size measured in all species compared to the reproductive population size indicates that forces are eroding genetic diversity much more than neutralist models predict. This erosion is due in part still poorly estimated to natural selection, which eliminates harmful mutations and fixes advantageous variations and thus increases the effect of drift on the neutral variation. Although extremely important for the future of the species, the selected polymorphisms certainly represent only a small fraction of the cases of polymorphism. Neutral molecular polymorphism provides the basic theory, the reference model from which the selection and history of populations are studied. The paradox is that from now on, the molecular signatures of natural selection are sought in the genome using neutralist theory. The existence of selective forces that maintain the recombination system, Mendel's laws, and the genetic mixing of sexuality is an argument for considering that polymorphism, which they maintain is this way, has a short-term advantage in natural populations. Dear students, it was all about today's lecture regarding introduction to ecological genetics and polymorphisms. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.